Hello everyone. I think I finally got all the kinks worked out with Panopto and how to integrate it into Blackboard. I again apologize for any missed lectures and that they were not there when they should have been. Well, they were there, but for some reason Blackboard doesn't think you should have access to things that I post, and so I figured out how to fix that. So again, I posted this in the announcements, but we'll make sure that you don't miss any points. There's nothing going on. You have maybe do a little bit of catch up. Hopefully you're getting there and you're caught up. I have made quiz number 5 available, and it will be available all today and until Friday at the end of the day. So that's just kind of as regular. Quiz number 4, which you didn't have the material for, I've also made available to the end of the day Friday. And also for quiz number 4, you can take it as many times as you need to. So you can retake that again and again if you need to do that. And then finally, the worksheet, which was going to be due this evening, that's Thursday, July 25th. I will extend that one more day, and we'll have you turn that in anytime by the end of the day Friday, and it won't be counted late. In addition, in this folder, also in addition to the PowerPoint for this unit 2.4, I also have 2 other documents that are overviews. One of the worksheet, talking about the concepts on the worksheet, going through its problem by problem, so you can take a look at that if you need some guidance on the worksheet. The other video there is a video on Hardy-Weinberg equations with some practice and some review. It's one of those things that sometimes is a little tricky to do the first couple times, but you practice it a little bit and go over it, and then you'll have a good chance to do that. In addition, there is a video, I guess you call it a challenge, that's also in this folder. So a total of 4 different things in this folder. 2 of them are optional but will be very, very helpful. One to help you on the worksheet, one to help you practice Hardy-Weinberg. And then the other one is, the other additional one is for that challenge, which you do for an attendance grade. So just take a minute and make sure you get those done. Okay, so this discussion is about looking at population genetics and how we can measure the genetic diversity of a population and then use it to determine whether or not there are evolutionary forces that are acting on that population. And you're going to see things that look a little bit like a Punnett square. So you've probably done Punnett squares before. So here's an example of a simple Punnett square where they're using A1 to represent one allele, A2 to represent the other allele. And when you did this, you were trying to predict what the offspring of 2 parents would be like. Now, 2 individuals, one whose last name was Hardy and one who was Weinberg, thus the name for this approach, realized that you could also do this at the level of an entire population. And this is something that came about because of Mendel and Mendel's work. When Mendel determined that you could treat populations or the alleles inside populations as individual particles, right? So the genes don't mix together. They stay independent. The extension of that is we also don't need to treat them as if they are stuck to the other allele. They act as if they are their own individual unit, not connected to the allele that's found just right across from them during meiosis. And that's because of this process of random assortment, okay? And so we can treat them as if we're sampling, especially if the population is very, very large. We can treat them as if we're sampling from an infinite population, and then we can use it simple probability predictions to predict what we should have as an outcome. So we're going to look at how, the answer to this question at the bottom, can we make predictions on a population level? The answer is yes, and I'll show you how we do that. Okay, so all we have to do is take this simple idea of Punnett square, which is simply probability, right? So the probability of getting A1, which is 50% of the alleles in this parent on the top, and the other A1 together, which is 50% of the alleles, is simply the union. That's the probability term, and it's just the product. It's A1 times A1. And so if we have 25% of the population, 25% of the alleles that are A1, then it would be 25 times 25. Now, if we're just doing 2 parents, then we know that 50% of the gametes carry A1 and 50% carry A2. So we do 0.5 times 0.5, and we end up with a result of 25% for the homozygous, 25% for the other homozygous, 25% for either of the combinations of heterozygotes, which gives us a total of 50% for the heterozygotes. And those are based on the simple laws of probability. Now we can extrapolate this to populations. And so we can move from a qualitative measurement, which means we kind of assess general things that are going on, but we don't know exactly how much or how fast. But we can begin to move from qualitative assessments to actually quantitative assessments. And quantitative assessments allow us to measure things much, much more precisely. And they also allow us to make extrapolations or predict what we should see in the future. So we can predict 1, 2, or 3, maybe even more generations ahead of time if we make some assumptions about the environment. And so we'll look at that and how we can do that. All right, so again, this is based on basic probability calculations. And there are two types of probability calculations. The probability of sample number 1 and sample number 2 happening at the same time is simply the product of those two probabilities. So if you have flipping a coin, for instance, the probability of in coin flip number 1 getting heads and in coin flip number 2 getting heads is whatever the probability is. If it's a fair coin, it would be 50% times 50%. You would have a 25% chance of getting 2 heads in a row. Okay? So the probability of getting in a single event, right? What are the probability that I get heads or tails? Now again, if those are the only two options and it's 50% for each, then the probability becomes 1. If you are flipping a coin, you're going to get either heads or tails. So just recognize that we're mainly going to look at this first one, which is called the union of probabilities, and we'll look at how that works. Now, to be able to do this for populations, there are two numbers that you need to be aware of, and you need to make sure that you don't get mixed up. The first is the genotype frequency. So just remember what a genotype is. A genotype for a diploid organism, which is all we're going to be dealing with for these examples, is the combination of the two alleles that they carry. So we can have homozygous genotype, we could have a heterozygous genotype, and then we could have the third possibility, the other homozygous genotype. Okay? Now, for many organisms, there are more than 2 alleles for each gene, and that's very, very common. But for illustration purposes, we don't need to get to those more complicated examples. So we're always going to look at these 2 allele systems. And sometimes you'll see them A1 and A2 representing the 2 alleles. Other times you'll see big A representing one allele and little a representing the other allele. Either way works. It's just convention and what's common. So we're going to go from a population of parents and predict what the gametes should be. And we can do this because, remember, A1 and A2, even though this is a heterozygote, those two things are not stuck together and inherited to their offspring. It's an either or, it's a randomly assorted thing. And so we can treat this A1 and A2 from that heterozygote as independent probabilities. And so the chances of that A1 getting paired with another A1 or that A1 getting paired with an A2 simply is the product of the probabilities that we find in the entire population. And so we can predict what we should see in the offspring. Now, in this example, if you'll notice, the offspring look a little bit different. I think we've got 2 homozygous A1, but we've got 3 homozygous A1 here. And we've got 3 heterozygotes, but only 2 heterozygotes here. Oh no, I guess we still have 3. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6, 7. Oh, we have one extra member of the offspring population compared to the parents. Now, in small populations, because of random chance, the predictions don't work very well, and we'll look at that. That's actually one of the things that violates Hardy-Weinberg equation. So in a way, you can think of this like flipping coins, and it works as a fairly good analogy for many of these things. If you flip a coin, say 6 times, and I say, predict what the outcome is going to be. Well, if you know the coin was even, that you got 50% heads with each flip, a chance, and 50% tails, your prediction would be, I should get 3 heads and 3 tails. But you realize with such a small sample size, it's very likely that you're going to have random variation so that your prediction won't match the actual outcome. It's very possible, in fact even quite likely, that you might get 4 tails and 2 heads, or even 5 tails and only 1 heads, and that's not outside the realm of probability. If, however, we expanded that sample, so instead of flipping 5 times, if you flipped it 5,000 times, again, your prediction would be half heads and half tails, and you would be very, very close with just a little bit of variance from that prediction. Okay, so remember, genotypes is simply how many homozygotes for A1 we have in a population, how many homozygotes for A2, and how many heterozygotes we have in that population. Those are your genotype frequencies. So for our examples, there are always going to be 3 genotype frequencies. And at the beginning of each problem, you will be given the genotype frequencies. That's how we start with these problems. Once you're given the genotype frequencies, you need to be able to figure out the allele frequencies. And I'm going to walk you through 2 examples of how to do that, and then you'll have 2 or 3 additional examples on the other video that's posted in this folder. Okay? So we'll walk you through how to do that. Remember, the allele frequency is just what percentage or what frequency of the A1 allele is found in the entire population in that gamete pool. What percentage of the A2 allele, or what frequency. So we only have 2 allele frequencies, one for one allele and one for the other allele. Now, to make the math easy, we always express these frequencies, just that as frequencies, where 1 represents like 100% and 0 would represent 0%. So 0.5 would be 50% of the alleles, big A, and then A1, and then we would know that the other 50%, if we're in a 2 allele system, would be A2. Okay, so hopefully that's making some sense. It kind of fits a little bit into your head, but let's look at practically how we would do this. So let's say we go into a population and we count genotypes. Okay, so we have 250 of the homozygous dominant. So I'm using this to represent the big A here, to represent the dominant allele, and so we have 2 copies were homozygous. Here we have 500 heterozygotes and then 250 homozygous recessive. Okay. This is a very common example, and you'll probably see this example maybe on a quiz, maybe even on a test, but you'll probably see it over and over again. It's a good one to start with. Okay. Now, the frequency of that big A allele, which is our allele frequency, can be represented mathematically like this, with this little italic F and then the A in brackets, but more easily and simply, it is often represented by the letter P. Okay. The frequency for the other allele, remember we only have 2, is represented by Q. Those are the 2 numbers that were picked by the first people that did it. They became kind of the standard convention. They could just as easily be Z and Y or any other 2 symbols to stand for those. They're just there to help us remember them. And we're going to look at an equation where we use these integers to stand in for the allele frequencies. Okay? Now, to do this, we have to do some calculations. Okay? You have raw counts of the genotypes, but we need to figure out what's the frequency of the big A allele, represented here by P. What's the frequency of the little a, represented by Q? Okay, so the allele frequencies for these populations are simply 0.5 and 0.5. So let me show you how. Let me show you how I get to that. Okay, so I'm going to type in the math of how we get to 0.5 and 0.5 so you can see it as it's done. Okay. Okay, so you're not going to be able to see this till I get back to presentation mode, but we're going to do 2 times 250, and then we are going to add in 500 individuals when we are calculating that big A allele. I'll make this just a little bit smaller so we can fit it all on the slide here. Okay, so here you go. Here's how that math works. Oops, and I put a plus instead of a equals. Let me get an equals in there just so you can see it and it'll make sense to you. Okay, there we go. So here's how that math works. I did 2 times 250 because I want to count all of the alleles. Okay, so I do 2 times 250 because there are 250 individuals that carry the big A. So I've got 500 big A alleles represented in those 250 individuals. So that is here 2 times 250. For the heterozygotes, I have 500 individuals. And so I have 500 individuals that only carry 1 copy of that big A allele. So I'm now going to add in those 500 single copies that the heterozygotes carry. So this is fairly straightforward math. 2 times 250 is 500 plus 500. Now, if you notice, that does not equal 0.5, so I'll show you how we get there. So first, before we do that, I'm going to count up all of the little A alleles also. So I'm going to do this one down here. Now, this is a nice, easy example because it's symmetrical. So calculating the Q value, this frequency of the little A allele, is going to be the same as calculating it for the big A allele. And so here's how it looks. Again, the math doesn't quite work yet. Let me show you what happens. But we have the same numbers. 2 times 250 to count up these homozygotes. They carry 2 copies of the little A allele. And then 500 individuals, because the heterozygotes carry 1 copy of the little A allele. And then to get frequencies, all we want to do is divide by the total. So for each of these, let me just type it in here so you can see it. Our final number of alleles is going to be 1,000. Okay, and then I'm going to write beneath it what we do here. So we have 1,000 divided by 2,000, right? Because there are 2,000 total alleles. And this is what it's going to look like. Let me get it back up on the screen here for you. So here we go. The frequency of the big A allele, which we're going to represent by P, we add up all the alleles, we end up with a value of 1,000. We then take 1,000 and we divide it by 2,000 because that's the total number of alleles in the population, and we end up with 0.5. Okay? Now, we would have the exact same thing. Let me put it down there on the little a allele just so you can see it there. Sorry, PowerPoint sometimes is a little tricky to get it exactly the way you want it to look. Okay. Make all my last little changes here. Let's move this down a little bit so you can see it. And here we go. Okay. So here's how this math works. Now, if you're not quite following or you're not exactly sure, I'm going to run through it here one more time. We'll do one more example, and then you'll have examples on the other video here so you can practice, and I'll even give you a few that you can work out yourself with answers posted later to see if you've got the hang of it. So 2 times 250 to represent all the homozygous dominant. We add in 500 individuals, the heterozygotes, because they carry one copy of that dominant allele. We get a total of 1,000 big A alleles. We do the same thing for the little a allele, and this is nice and symmetrical, so it's easy. 2 times 250 to represent the homozygous recessive. They carry 2 copies, plus 500 to represent the 1 copy of that recessive allele that the heterozygotes carry. And we get a total, oops, I missed that. That should be right here, should be 1,000. We get that in. There we are, back to our presentation. So there we are, we get 1,000. We divide by the total. 1,000 and 1,000 is 2,000. So we divide each by the total, and this is where our final frequency of 0.5 and 0.5 comes. Okay? So why do we care? It seems like a long involved process. Why do we take our genotype frequencies and convert them into an allele frequency? Well, this is where the probability comes in. Hardy and Weinberg realized that if you knew these allele frequencies, if there were no evolutionary forces acting on a population, you could predict what the genotype frequencies should be in the population if everything is in balance. And if your predictions don't match your original observations, then you know that something is going on, that those populations are undergoing some sort of evolutionary pressure. There's some evolutionary force. So here's how that works. Okay, so in the next generation, our prediction matches what we call the Hardy-Weinberg equation. Okay, so let's get that up on the screen here. Oops, back here. So predict the frequency of genotypes. I keep clicking forward. Predict the frequency of genotypes in the next generation. So here's what we do. All we need to do is take our p-value, and I'll get it up here on the screen in just a second, and we do p squared. Okay? The q value is 2pq. And then finally, I said q value. The heterozygote prediction is 2pq. The prediction of the frequency for the genotypes of the homozygous recessive is q squared. Okay? So here is the final version of this. Now, I know this is a little complicated. You might not quite have your brain wrapped around it. You say, oh, this is math. I hate math. It is a little bit of math, but really it's not too bad. Once you do a couple examples, you'll start to get the feel for it, and it'll be pretty straightforward. So in the final version, this is the Hardy-Weinberg equation down here in the bottom right. P squared, which represents the predicted frequency for the homozygous dominant, plus 2pq, which is the predicted frequency for the heterozygotes, plus q squared, right? And so here are those just so you can see what they are associated with. Now, if we plug these numbers in here, our p-value is 0.5. So 0.5 squared is 0.25. So in other words, 25% of the population is predicted to be homozygous dominant if there are no evolutionary forces. And sure enough, when we look at the population from which these numbers came, we see that there are 250 out of 1,000, or 25%, that are homozygous dominant. So our prediction matches our original observation. Okay, next one, 2pq, which is 2 times 0.5 times 0.5, which, you do the math, that is 0.50, or 500 out of 1,000. Our prediction matches our initial observation. And then finally, Q squared, which is 0.5 times 0.5 or 0.25, again, matches our observation. Prediction matches the observation. Now, if you do your math right, this p squared plus 2pq plus q squared will always equal 1. So we're not trying to determine, that's one of the common mistakes students will make. They'll plug the numbers in and say, yep, it equals 1, everything's in Hardy-Weinberg equilibrium, boom. But that's not what we do. They're always going to equal 1 unless you've made some sort of mistake. What we're looking for is, does this first term, the p squared, match our observation? Okay, does this second term, the 2pq, match our observation for the heterozygotes, and so on. Okay, so let's do one more for the where we're given different genotype frequencies. Okay, so this is the one we're going to do. I'm going to pause it and type in the information, and then we'll go over it. Let's try it again. Okay, so here's what that looks like. We're going to do it the same way we did before. We're going to count up all of our homozygotes and times them by 2 because they carry 2 copies of that big A allele. We're going to add in our heterozygotes because they carry 1 copy of that big A allele. When we get them all up, we end up with a total of 400 alleles. We're not done yet. First, let's do the Q value. 2 times 640 because there are 640 individuals that carry 2 copies of the little A allele. We add in the heterozygotes because they also carry 1 copy of that little A allele. We get a total of 600 little A alleles. Sorry, 1600 little A alleles. We then divide by the total. Now 1600 and 400 is 2000, so we take 400, divide by the total to get a percentage or a frequency, 0.2. 1600 divided by 2000, 0.8. Now notice, if we've done this right, our numbers will always sum to 1. We then plug those into the equation, right? So the allele frequencies for these populations, if there's no evolution, are predicted by this equation. P squared, which is 0.2 squared, right? What is 0.2 times 0.2? Now, if you'd like a calculator, which you probably will want one unless you're some kind of a math whiz, which is great, we need math whizzes too, but if you're not, bring a calculator, right? So put it in your calculator, 0.2 times 0.2 is 0.04 or 4%. And notice 40 out of 1,000, which is 4%. So our prediction matches our observation, our original observation. 2pq, which is 2 times 0.2 times 0.8, which you put the math in, you do it in your calculator, you're going to get 0.32 or 32%. Sure enough, it matches. And then lastly, 0.8 squared, right, which is our q squared down here, is 0.64 or 640 out of 1,000. So again, all of our predictions matched our end observation. So we know there's no evolutionary force acting on this population. And if that's true, it's said to be at Hardy-Weinberg equilibrium. Now, that's not always going to be the case, but scientists use this as a quantitative measure to determine if there is evolution acting on a population. And if your final predictions don't match your original observation, that means there is evolution acting on the population. And sometimes scientists will even use this to predict what would happen in the future. So if we added a natural selection force, or if there was a bottleneck where the population suddenly got really, really small, we may be able in some cases to predict or at least give a general overview of the range of outcomes that we might expect doing that. Now, I'm going to jump ahead to this one. There are many, many places where populations can be at Hardy-Weinberg equilibrium. They can be at 50-50, like we saw in the first example. They can be at 20-80, like we saw in the second example. So in a way, mathematically, the way this equation looks when you graph it out is kind of like a half pipe. And just like a half pipe, there are multiple places of balance. If you throw a marble into a half pipe and I ask, hey, where's it going to end up? If the half pipe is perfectly level, you'll say, well, it's at the bottom somewhere, but I don't know if it's in the bottom in the middle, or in the bottom at this end, or the bottom over there. It's going to end up at the bottom. That's the point of rest or equilibrium. Okay. Now, there are 5 forces that violate Hardy-Weinberg equilibrium. So if we go through this equation, which you'll see on the first example, that's also posted in the same folder. So look at that as an example of something that's not in balance. But if we find our predictions not matching our original observations, then we know one of these 5 forces is acting on a population. So natural selection acting on a population will violate this null model, this Hardy-Weinberg equilibrium. If there is non-random mating, that means if individuals are choosing a mate based on the frequencies that you're measuring. So if like mates with like, or individuals choose a mate who looks very, very different from themselves for this genotype you're measuring, then we're going to see things out of equilibrium. if there are new mutations that are frequently coming into the population. And we're going to talk about each of these in turn here in just a little bit. If there's migration from a population with a different genotype frequency, that can throw off our overall allele frequencies. And then finally, small population sizes. And the reason is small population size will throw off our frequency is the same reason we talked about earlier. If we only flip a coin 5 times, we have a very unpredictable outcome with a small population size. And that's actually the same force we've already talked about called genetic drift. And genetic drift is most strong in small populations. So if you look at these, we basically have the big three we talked about at the beginning of class, right? Natural selection, mutation, and then genetic drift here in the form of small population size. And we've added 2 more. Now some people put both migration and non-random mating as natural selection forces because they're forces on the environment that can have an impact on the fitness of an organism. Some people will classify them that way, but for Hardy-Weinberg purposes, we separate them out. So you should know these 5 as forces that can violate this nice balance. So when a population is not undergoing evolution, it will be at one of the balance points. Okay, now again, take a look at the little discussion in the video here that will have Hardy-Weinberg equations for you to practice, some that are out of balance, and I'll go through it. I'll put in the numbers. I'll kind of type it out bit by bit as you go through and explain what I'm doing. I will then also have a practice there for you to look at and for you to practice on your own and give you some time to do that. Okay, so practice there. We'll get a little bit of practice in class on Friday. There's a little bit on the worksheet, and we'll see some of those problems pop up there also. So let's take a look now at these 5 forces. We've talked about natural selection already, so we're going to spend some time on numbers 2, 3, and 4. We'll save genetic drift and some more details about genetic drift for a later date. Okay, but let's look at the effect of mutation on Hardy-Weinberg equilibrium. Now, we talked about mutation as an essentially random event, and it really is. So the prediction, the effect of mutation on Hardy-Weinberg is very, very difficult to predict, and usually it's not very great, because even though mutations are fairly common, the chances of them happening in one particular gene out of the many thousands of genes that organisms carry is fairly low. And so for the most part, we can ignore the effect of mutation on Hardy-Weinberg. If it does occur, if it is a real force, it's very unpredictable. Occasionally, we can look at the effect of mutation as a loss of function, because there can be many different mutations that give the same loss of function. So for instance, you sometimes have individuals that are born that are blind, that have a genetic mutation that makes them unable to see. And there could be many, many different mutations in the many genes that help develop and control eyesight that could cause an individual to be blind. And so sometimes people will look at that effect of mutation on Hardy-Weinberg. And so in general, when we're looking at the entire genome, the number of deleterious alleles in the population are going to reach a balance. And we basically have 2 forces. We have forces bringing in deleterious alleles, negative mutations, right? And that's just mutations during meiosis. Those bring in negative alleles at some frequency. Okay, and we talked about kind of a ballpark of general types of frequencies that we see in discussion 2.1. And then we also have forces that remove these deleterious alleles. Natural selection is a big one, right? So natural selection removes these negative alleles. It works to anything that's bad. Those organisms don't reproduce. They don't pass on their genes, or at least not as much. And over time, those negative alleles, deleterious alleles, are removed from a population. So in short, deleterious alleles reach a balance that depends on the mutation rate, which is bringing in new deleterious alleles, and the force of natural selection, which is removing those. Okay? Now, migration, again, oftentimes we have very different populations that can be located close or maybe somewhat distant to one another. Here's we have an example of a mainland and then a population on an island that has a different genotype frequency than on the mainland. So if we had frequent one-way migration, if there was a current or some other natural force caused organisms from the mainland to more frequently come over to the island, over time that would change the starting frequency on the island to eventually it would reach a balance point, and so that could have an effect. Now, I'm going to give you a little bit more of an in-depth definition of population. Up until now, we've been treating populations and species as essentially the same thing, which is okay, but realize that that's an oversimplification. A species is a population. However, it's also possible and often very helpful for scientists to divide species into subpopulations. And so we could think of this island group of organisms as one population, and the island group over here, and the mainland group as a second population. And so populations can be species, but not all populations are species. Sometimes we have subdivisions. So we might have a subspecies, or we might have something that we call a variant, or in human populations, sometimes we say a race, or maybe even better, an ethnicity, right? Those are subpopulations that, because of some past historical events, we see some distinctions, whether it's a cultural distinction or some sort of a genetic distinction. We see that based on past history of those populations. So just be aware of that. Now a little bit more detailed, and I guess perhaps complicated, but a better idea of what the definition of a population is. Now, non-random mating occurs when individuals choose a mate that is either like themselves, which would be assortative mating. So if orange butterflies prefer to mate with other orange butterflies, that would be assortative mating. Disassortative mating is where individuals will preferentially avoid individuals that look like themselves. And in natural populations, we find many examples of both. It just depends on the species. And we can actually see this probably a little bit in humans, right? So especially in the past, there was some strong assortative mating within certain cultures, where individuals were much, much more likely to mate with an individual from their own culture than somebody from another culture, and even sometimes when they were in close proximity. So for instance, during the early 20th century, many Jewish people migrated to America, and they set up communities in large cities, and sometimes in other areas, but primarily in large cities in the United States. But despite that, many of them continued to stay fairly insular, and they would look for mates or get mates chosen for them by their parents that were from that same community. And so there was some amount of assortative mating. Strong assortative mating is often also called inbreeding. And if it gets too pronounced, if you're mating with individuals that are too much like you, there are some downsides to that. It's detrimental. Okay? Disassortative mating, which we also see in human populations, there is nearly a universal taboo against mating with a close relative, like a sister or a brother or a mother or a father. Those in nearly every human society are really looked down upon and strongly discouraged by our society. And so that is a form of disassortative mating. It prevents individuals that are very closely related to one another to breed and have offspring. Okay, so we see examples of both in different human cultures. Now, the strongest type of inbreeding is self-fertilization. This happens actually quite commonly in some plants, but even in plants, usually there are mechanisms to limit the amount of self-fertilization, although there are some plants that do a large amount of that. The downside of mating with close relatives, and again, the very closest relative to you is yourself. Most animals, even those that are hermaphrodites that carry both male and female reproductive structures, most animals are unable to self-fertilize, and I guess even more strongly, most animals are have males and females in different organisms. A single individual does not carry functional male and female reproductive organs. There are some exceptions to that. Gastropods are a good example, the snails, where some species have both male and female reproductive parts. However, it's rare for them to be able to mate with themselves. So inbreeding is something that's common in plants, and we see it here and there across other groups. But plants, it's a big phenomenon and force that we see in them. Mating with close relatives is negative because of something we call inbreeding depression. Now, the main downside, the main thing that causes inbreeding depression, is the pairing of rare recessive alleles. It's estimated that on average, every human carries one or two alleles that are very detrimental, maybe even fatal. Now, hopefully this doesn't freak you out. It shouldn't. But if there's a good chance that you carry a fatal allele. You say, well, wait a minute. Fatal means dead. I'm not dead. What's going on? Well, it's recessive. And so you have a dominant allele from one parent and you got a recessive allele from another parent. And so you're a carrier for that gene, but it doesn't affect you phenotypically. And the only way it can impact your fitness is if your mate also carries a copy of that rare recessive allele. And so if we're looking at tens of thousands of genes in the human genome, and you have a random one that's got a defective copy, what are the chances that your mate also carries that rare recessive allele? And so, of course, it depends on how rare it really is. But if your mate is very, very distantly related to you, then it's almost very low chance that, or essentially close to zero chance that they carry that allele. But sometimes it happens, and if you mate with close relatives, it has a much higher level. And so we actually see the downside of this in human societies that have had inbreeding going on for many, many generations, and the Jewish societies that establish themselves in the big cities in the eastern United States are actually an example of this. There's a fairly high level of a recessive genetic condition called Tay-Sachs disease that pops up among those Jewish populations. And it's not that they had defective genes. Any population that did that, there's another kind of famous example. There were some British sailors and Polynesian women that were marooned on a very, very remote island in the South Pacific. And they had a couple generations of time, and there's a very high level of a very rare recessive disease that has popped up in that population. It's just whatever random recessive alleles were carried into that population. It's inevitable that it's going to happen if you have enough inbreeding in the population. Most species have some form of disassortative mating. So in these birds, there's strong evidence that males, which show some diversity, tend to choose females that look different from themselves. So in the past, that was an advantage. It was just a random mutation that popped up on what males prefer and what females prefer, and the ones that got an advantage from that did better, and so we see that perpetuated in the species. So here's a little box where they're doing an experiment. They put a female bird in. They're asking her to choose a male that looks different from her or a male that looks the same. And on average, she'll choose the male that looks different from her more frequently than the one that looks the same. That's a disassortative mating. We see lots of this in other species. One other last example is MHC, which stands for Major Histocompatibility Complex. And mice, if given a choice, will choose a mate that has a different MHC than themselves. And MHC plays a role in immunity. And the more diverse your MHC, your major histocompatibility complexes are, the better you do. And so in the past, mice that were able to choose a mate that had some diversity and variety for those did better because their offspring inherited a little bit of a stronger immune system. Now, how does a mouse do this? Well, it's obviously some sort of a chemical cue, but I don't know how a mouse chooses a mate. But many mammals do this, where they will size up visually. They will get chemical cues by smelling, maybe even touching and physical cues that might convey some sort of information on choosing a mate. And so mice are able to do that and assess that, and that creates diversity among offspring and has been a grand advantage for many of those mice. And so we see this in many species. Okay. That takes care of discussion 2.4. On the syllabus, we had 2.5 that we were going to discuss and go over today also. But I think because of the technical snafu and because we got a little bit delayed, we'll skip 2.5 for now. We'll just push it back. I guess not skip it. And we'll do 2.5 on Friday and do a little bit of 2.6. And that will help make up and even out the week for you so you don't have quite so much to cram into the last week. Now remember, coming up on Monday, so you still have a few days, but coming up on Monday, you have an exam number 2. So we'll spend Friday doing the last little bit of material from unit number 2. We'll do review. You've got a worksheet due at the end of the day on Friday. Remember that. You also have 2 other videos in this same folder. One looking at, I guess 3 other videos, sorry. One looking at the worksheet, little help pointers on the worksheet. One looking at practice for Hardy-Weinberg equation. And the other one, which is one you need to look on, there'll be one short question at the end, a little survey type thing. You get full credit whether you get it right or wrong, but for you to do for basically an attendance grade. It's like a remote digital attendance grade, I guess. So anyway, again, good luck with that. Remember, practice Hardy-Weinberg, go through it, a couple examples on that worksheet. You'll get comfortable with it. If you're not, you're welcome to come by my office. We'll be doing some in class also. So good luck, and I will see you on Friday at 11:20.